So our DNA, DNA structure, DNA stands for deoxyribonucleic acid. So in terms of organic molecules, it's a nucleic acid. And its function is to store and transmit hereditary information. We'll leave what that means for another time, exactly what type of information we're talking about. But today I want to focus on what the structure is. And the structure of DNA wasn't really known until the early 1950s. And its final structure, although credited to a small number, was really due to quite a few different leading discoveries up to it. Starting with knowing that they should be looking at nucleic acids for as the model of heredity. At one point in time, there was question as to whether it was proteins or nucleic acids. And Hershey and Chase had a pretty famous experiment that showed it was an example that nucleic acids are that molecule of heredity. And then Watson and Crick are well known as they received the Nobel Prize for the discovery of DNA. And just to add a little bit of intrigue in there, they used an image captured by Rosalind Franklin, who was a female scientist at that time in a world of men. She wasn't particularly well liked, whether from her own personality or the fact that others, the men in the field, didn't think she belonged there. But she had a really pivotal role in the discovery with some X-ray crystallography photographs that she took. So there are a couple videos I'd suggest. I'll put them down in the bio underneath. In terms of the history of discovery and what role did Watson Crick and Rosalind Franklin play. But today the focus is on the structure of DNA. So like other organic molecules or macromolecules, it's got a building block, and the building block of DNA is actually called the nucleotide. And a nucleotide is made up of three parts. It's made of a phosphate group connected to a sugar. This is our sugar, and then to some type of nitrogen base. All right, so this is our nucleotide. And you'll notice that I put the phosphate up here. That's where the phosphate belongs in the sugar, with this upwards stretching arm of the sugar. And if we actually look a little bit more closely at the sugar, we're going to find that the sugar is put together with a carbon ring structure that looks a little bit like this. Okay, we'll come back to that later. So these nucleotides are what's assembled into DNA. And when DNA is being built, it's the whole nucleotide that's being put in. Somewhere else in the cell, nucleotides are built. And then when it's time to assemble DNA, the nucleotides come together and form DNA by bringing themselves in, they're attached, and you end up, if we just did one line of these, attaching nucleotide after nucleotide, we'd end up with a chain of nucleotides, all attached to each other in the same way. All right. And each of these is going to attach to another nucleotide on the other side. We'll draw a couple of these in. Where our phosphates are connecting our sugars together, and across the way between nitrogen bases, we are going to have hydrogen bonds holding our nitrogen bases on, and then we have stronger bonds holding our sugar phosphate backbone in place. Now, nitrogen bases are specific. They have specific pairs in which a purine is always going to bind with a pyrimidine. And beyond that, they are more specific than that, as the purines and pyrimidine matches are going to be specific adenine pairs with thymine and guanine pairs with cytosine. And that has to do with the number of hydrogen bonds that they form between each other. So when you're trying to remember these, adenine and thymine go together, and guanine and cytosine. Mnemonic devices are helpful. So apples grow on trees. There's my apple. There's my tree. And this one is that cars will park in, not a very good car, but we'll drive a car here. And cars park in garages. There are other mnemonic devices out there, but adenine always pairs with thymine, guanine always pairs with cytosine in DNA. So these purines versus pyrimidines, then, to take a moment, a purine is always going to have a double ring structure. So our purines are made of 2 carbon rings connected together. So adenine and guanine have this 2-ring structure, and our pyrimidines then are going to have a single ring structure. And I put them in order with them. So your pyrimidines are going to be thymine and cytosine. And I think the easiest way to remember that is that they both have a Y's. And your purines then are going to have 2-ring structure, and they're adenine and guanine. All right, so let's go back to our molecule up here, and let's identify for a moment a nucleotide. A nucleotide is going to be 1 sugar, 1 phosphate, and 1 nitrogen base. So this guy right here would be an example of a nucleotide. Now, our nucleotides are connected to each other, and sometimes you'll hear a reference to a sugar-phosphate backbone. The sugar-phosphate backbone is the backbone of connected sugars and phosphates. Every once in a while, you'll hear DNA referred to as a ladder, and that ladder, the sugar-phosphate backbone, would be the sides of the ladder, while your nitrogen bases would be the rings. So these are both nitrogen bases. And holding up, not rings, but rungs, holding the nitrogen bases together, you will have these dotted lines represent hydrogen bonds. And one more piece here, and you'll notice that I drew one side of the DNA going one direction and the other side going the other. DNA forms these parallel strands that are bound to each other. And although the strands are parallel, they are not identical strands. They run in opposite chemical directions. And so those chemical directions actually come from this sugar up here. So I'm going to go back to my original sugar drawing and point out that our oxygen is at the peak of our pentagon-shaped ring. And each carbon after, we can count 1, 2, 3, 4, 5 carbons. Though the carbon number going in that clockwise order is going to give you the direction that DNA is facing. If you notice that I'm always drawing the phosphate on this upper side, the 5' side, so the only open space is underneath. So when nucleotides are put in, they're put in with the phosphate attached to your 5' sugar, your fifth carbon in the grouping. So then that is going to tell us the direction of this DNA, and we're going to call that the 5' end of the molecule, and down here we'll call this one the 3' end of the molecule to represent the 3 carbon. The other side of the molecule, if you look how I've drawn this sugar, I attempted to draw it upside down. So our phosphate is off of our 5th sugar facing the opposite direction. So that means that our 5' direction of our second strand is across the 3' section of our first strand. And that leaves your 3' for the upper end. This is important in DNA when we start to talk about how it functions, because The only space that's available to add on a nucleotide is right here on the 3' end, where there is no phosphate currently bound. On the 5' end, you already have a phosphate. So if you're talking about building in new nucleotides, you're not going to bring a nucleotide to this phosphate and try and attach it here. You're going to bring it in where the phosphate can attach to this open space in the sugar and form the new bond there.